Can somebody please confirm you can hear me?
I can hear you.
Thank you. What's there today? All right, let me share my screen. Which one's changed too? Okay, today is May 2nd, and we are supposed to be finishing chapter 4 today. There will be a lab today. Any questions about what we're doing? I'm not sure we'll finish chapter 4, but we'll try. All right, I have that we were talking about the fluid mosaic model of the plasma membrane, where the plasma membrane is actually a fluid. And I think I pointed out my hand and said it's held together by cells that are held together by the cell membrane. And it's amazing when I think about it that my hand is held together by a fluid. The plasma membrane shows selective permeability. It allows the passage of some molecules, but not other molecules. No large molecules can move across the plasma membrane. Did we talk about this?
I think so a little bit.
All right, maybe we got started on this slide and I went back to the slide we finished. Ions cannot move across the cell membrane, or if they can, they move very slowly. Lipids can move across the cell membrane because lipids can dissolve in the cell membrane. However, there aren't too many lipids outside the cell. If you think about like a red blood cell in the blood, there are some lipids in the blood, but most of the blood is water and molecules dissolved in the water. Smaller molecules like oxygen and carbon dioxide may diffuse across the cell membrane. The plasma membrane can contain, I don't know, different components, different features. For example, in prokaryotes, it contains the enzymes for making ATP. If the prokaryote is, or a bacteria, actually it's a prokaryote, is photosynthetic, it will contain the photosynthetic pigments. The photosynthetic pigments are found in the foldings of the cell membrane, and the foldings are called either chromatophores or thylakoids. Excuse me, my voice has gotten worse all of a sudden. Let me blow this up. So here we have the cell membrane. The cell wall is on the outside, but the cell membrane in this cell isn't going along like that, along the inside of the cell wall. The cell membrane is actually going like this, and then going around, then going like that, and then going around. So these are the, in this case, chromatophores, which contain the photosynthetic pigments of a photosynthetic prokaryote. If you damage the plasma membrane, you'll have leakage of the cell contents out of the cell, and that will kill the cell if the cytoplasm leaks out of the cell. You can damage the cell membrane with alcohols, quaternary ammonium compounds, and with polymyxin antibiotics. All of those put holes in the cell membrane. You have movement of material across the membranes. For a eukaryote cell, it's across all membranes, but for a prokaryote, it's only across the cell membrane. The movement of material across the plasma membrane is critical to cells. Cells have two processes used for moving material across the membrane. There's passive processes and active process. Passive process is where a molecule moves down its concentration gradient, moving from an area of high concentration to an area of lower concentration. In the passive process, you can have three of them. There's simple diffusion, where a solute simply diffuses across the cell membrane from areas of high concentration to areas of low concentration. And that's how oxygen and carbon dioxide move across the cell membrane. There's also facilitated diffusion, where a solute moves from areas of high concentration through a protein across the membrane into, well, either into or out of the cell. And that's called facilitated diffusion. There's also osmosis, where water moves across the membrane. Water is a solvent. It is not a solute. And osmosis always involves at least a semi-permeable membrane. Osmosis in cells occurs across the cell membrane. Any questions about that? So osmosis is similar to the other two. It's just that it's only involving water, which is the solvent, whereas the other two involve solutes, solutions, chemicals dissolved in the solvent. Cells also have active processes where the molecule moves against its concentration gradient, meaning a molecule moves from low concentration outside the cell to inside the cell, or it could move from low concentration inside the cell to outside the cell. The movement against its concentration gradient requires energy, and this happens by a transport protein, and the transport protein gets the energy it needs to move that molecule against its concentration gradient from ATP. Simple diffusion is just the movement of a solid from areas of high concentration to areas of low concentration. You'll note simple diffusion does not require a membrane. It can occur across the membrane, like oxygen and carbon dioxide move across the cell membrane, but putting a drop of dye in the water, the dye will diffuse from areas of high concentration to areas of low concentration. And obviously that's not across the membrane. So simple diffusion may occur across the membrane, like oxygen, but it does not require moving against the membrane or across the membrane. Any questions about any of that? All right, facilitated diffusion is similar to simple diffusion, but facilitated diffusion always involves a membrane. It has a protein that allows the molecule to move across the membrane. And this is called a transporter protein. It may be a transport protein or it may be a channel protein. It's the substance moves from areas of high concentration to areas of low concentration. And glucose is not a good example, so let me use a better example, and that would be fructose. Moves into the cell by facilitated diffusion, moving through a transporter protein, and fructose moves only from areas of high concentration to areas of low concentration. Like when you eat a lot of fruit, you have a lot of fructose in your gut, and that fructose will move into the cells by facilitated diffusion. That's the only way that fructose moves into your cells. Any question about any of that? What does that mean? It means that not all of the fructose will ever be dissolved, because once the concentration of fructose inside the cell equals the concentration outside the cell, no more fructose can move into the cell. And I should say there's no net gain of fructose moving into the cell, because for every fructose moving in, there will be one moving out. That's different from glucose, which is why glucose is not a good example. When glucose is higher at concentration outside the cell than inside the cell, glucose will move in to the cell by facilitated diffusion. However, when the glucose concentration outside the cell equals the glucose concentration inside the cell, this transporter protein will change from facilitated diffusion to active diffusion, and then it'll act like a transport protein, pumping the glucose from areas of low concentration outside the cell to areas of high concentration inside the cell. Everyone catch that? So the transporter protein, in the case of glucose, actually changes its function. Initially, it acts as a facilitated diffusion whenever the glucose is higher outside the cell than inside the cell. But once the glucose concentration outside the cell equals the glucose concentration inside the cell, the transporter protein changes to an active transport protein, and then will pump every one of the glucose molecules outside the cell to inside the cell. And that means you will digest all the glucose. Any question about any of that? We'll talk about active transport in a little bit. And I won't test you on glucose, the movement of glucose into the cell. I might test you on the movement of fructose into the cell, but I won't test you on glucose, okay? Because it's a little complicated. In the both cases of simple and facilitated diffusion, solutes are transported, and the solutes move down their concentration gradient from areas of high concentration to areas of low concentration. Osmosis is similar but a little different than both simple and facilitated diffusion in that it involves the movement of water. Water is a solvent, not a solute. And osmosis always involves a selectively permeable membrane. You do not have osmosis without a semi-permeable membrane. The same thing can be said with facilitated diffusion. You always have a selectively permeable membrane with facilitated diffusion. Water always moves from areas of high concentration to areas of low concentration. Now you should note that although it's water moving from areas of high concentration to areas of low concentration, we define the tonicity not by the concentration of water, but by the concentration of the solute. So the tonicity looks at sort of the reverse of the water concentration. And that is wherever water concentration is high, solute concentration will be low. And wherever water concentration is low, solute concentration will be high. Any question about any of that? So a bacterial cell, or actually any cell, can experience one of three kinds of osmotic solutions. There's an isotonic solution, a hypotonic solution, and a hypertonic solution. Each of these are relative terms, so it's correct to say the solution outside the cell was hypertonic relative to the solution inside the cell. Isotonic is the easiest. That's where the concentration of solute outside the cell is equal to the concentration of solute inside the cell. That means there's no net movement of water into or out of the cell. By no net movement, I mean if one water molecule moves into the cell, then one water molecule moves out of the cell. So effectively, there's no net movement of water. Cells are happy in this solution. They don't go through osmotic lysis. Any question about any of that? Like all of your cells are in an isotonic solution, and it has to be that way because animal cells do not have a cell wall to protect the cell from either a hypertonic solution or an isotonic solution. Now, it is true we do have a skin, and the skin does protect us for a long period of time. Like when you go swimming in the swimming pool, your cells are not going to burst from absorbing the water. And that's because our skin, will slow down the uptake of water into the body. I'm not sure. Eventually, though, enough water would move into the cells to make the cells start to burst. It must be a long period of time, though, because I've never heard of anyone dying from being in the swimming pool. But like I said, our skin would protect us from jumping in the swimming pool. We do have some movement of water into the body, but it's very slow. And I think you know this, like when you are in the pool for a long period of time, or you're in the bathtub for a long period of time, and your fingers turn kind of what we call webbing. A hypertonic solution is where the solute concentration is lower outside the cell than inside the cell. That means there's more water outside the cell than inside the cell. So the water moves from outside the cell to inside the cell. This will swell the cell membrane as water moves into the cell. If the cell has a cell wall, the cell membrane can only expand up to the size of the cell wall. And then the cell wall prevents the cell membrane from expanding any further, and that will limit water from moving into the cell. No more water, no net movement of water will move into the cell because there's no more room for the water molecules inside the cell. It is dangerous for a cell if the cell wall is damaged or if there is no cell wall, such as when the enzyme lysozyme or penicillin, the antibiotic penicillin, damages the cell wall. The cell will then have water continue to move into the cell, and the cell membrane will expand out of the damage in the cell wall, until the cell membrane bursts, and then that will leak the cytoplasm outside the cell and the cell will die. Most bacteria live in a hypotonic or an isotonic solution. When you have bacterial infection, the reason why penicillin G works is because the bacteria will be in a hypotonic solution inside you. And that's why penicillin works. You damage the cell wall, then water will move into the bacteria until the bacteria bursts because of the damage to the cell wall from penicillin. Any question about any of that? When the cell wall bursts, that is called osmotic lysis. A hypertonic solution is when the solute concentration outside the cell is higher than the solute concentration inside the cell. The net movement of water will be from inside the cell moving to outside the cell. This will lead to shrinkage of the cell membrane. It'll pull away from the cell wall and then start shrinking up. This can lead to plasmolysis, and generally it leads to cell death, depending on what cell we're talking about, like an endospore will not be killed by this. The bacteria, Mycobacterium, will not be killed by that, but most bacteria will be killed when they fall into a hypertonic solution. An example of a case like that would be a bacteria falling into molasses. The water will leave the bacteria and kill the bacteria. And that's why molasses can stay at room temperature for years. My mother actually had a bottle of molasses that we figured was at least 20 years old, and I made molasses candy out of it and fed it to my father, who really enjoyed it. Didn't kill him or anything like that. That bottle actually was at least 30 years old before my mother finally decided maybe it's time to throw this bottle away. And I said, well, you might as well because we haven't used it for 10 years. And actually for 30 years, but my mother used it, I don't know, 30 years ago, and then just didn't use it any further. All right, active transport is great when the concentration of the molecules the cell needs is higher outside the cell than inside the cell. That's not active transport, but passive processes are okay when the concentration of substance required by the cell is higher outside the cell than inside the cell. But what about when a cell is in an environment with low nutrient concentration, such as a cell in the ocean or a cell in the desert, like the Sahara Desert? What's the cell to do then? The cell then must rely on active transport to move the molecule from areas of low concentration to areas of higher concentration to move it inside the cell. To move a molecule against its concentration gradient requires energy. The cell needs to perform work to move the molecule against its concentration gradient. Cells use active transport to pump the solute molecules against its concentration gradient. Active transport requires a transport protein that's in the cell membrane that pumps the molecule against its concentration gradient. We're going to say moving it into the cell against its concentration gradient. Generally, transport proteins are molecule specific, meaning we have an active transport protein for glucose and we have one for galactose, and we have one for lactose. Any question about any of that? You can have a drug that can mimic a molecule and be transported by the active transport protein. But other than that, transport proteins are generally molecule specific. So we'll talk about them as being specific for one molecule. The reason why the transport protein can bump the molecule against its concentration gradient is because to move one molecule against its concentration gradient through the transport protein requires one ATP. The transport protein gets the energy it needs to pump the molecule from ATP. Any question about any of that? So for each molecule you move into the cell, you lose one ATP. Group translocation is a special form of active transport only found in prokaryotes. Group translocation is similar to active transport in that it does require a transport protein and that a molecule is transported from areas of low concentration to areas of high concentration across a semi-permeable membrane, which in a prokaryote will be the cell membrane. A high energy phosphate compound is used. It's not usually ATP. It's another high energy phosphate compound like PEP. You don't really need to know what PEP is, but we're just going to say this high energy phosphate compound gives up a phosphate group and releases that energy, which the transport protein uses to pump the molecule across the concentration gradient. An example would be glucose being transported across the cell membrane by group translocation, which is only going to happen in a prokaryote cell, because eukaryotes do not have group translocation. The transported molecule in group translocation is chemically altered. So the phosphate group comes off of PEP, and then it's transferred to glucose. So we have glucose outside the cell, and when it's transported into the cell, glucose is changed to glucose 6-phosphate. Or maybe it's 1,6-phosphate, it doesn't matter. Glucose phosphate, we'll call it that. It's thought that the alteration of glucose helps prevent the escape of the glucose from the cell, and also it allows the concentration of glucose inside the cell to be lower than it would be inside the cell if the glucose was only moved by active transport. Let me try and explain that a little better. Let's say we had 10 glucose molecules. Oops, 10 glucose molecules outside the cell, and it wants to move inside the cell. And for reasons of simplicity, I'm just going to say there's zero glucose inside. I guess I should put outside there. Let me blow that up so you can see it better, especially with the coloring here. It's kind of hard to see. So, all right, if we only had active transport, this would go, let me just copy this. If we only had active transport, you would have, let's say we have one glucose outside the cell now and 9 glucose inside the cell. To move this one glucose into the cell will be really up against its concentration gradient, because that would be, I don't know, moving 1 close to 10. And that's a high concentration gradient to be moving that last glucose. If, on the other hand, we have both active transport and group translocation, oops, you can move 5 so that when you're moving that last glucose outside the cell, instead of going against a gradient of 9, it'll be either 4 or 5. Let's talk about translocation. And it won't be glucose inside the cell, it'll be glucose phosphate inside the cell. The point is, instead of being transported against close to 1 to 10, you'll be 1 to 4 or 1 to 5. Do you see that, everyone? That's a much lower gradient, 1 to 4 or 1 to 5, than 1 to 10. And it's easier moving the glucose into the cell when you have both active transport and group translocation to move the glucose into the cell. And prokaryotes can do this. And they do. Any question about any of that? All right. The cytoplasm is the liquid substance inside the cell membrane. It's mostly water. About 80% of the cytoplasm is water, but it does have dissolved solutes in it, like glucose and salt and magnesium and chloride and all those other things that are in the cytoplasm. The nucleoid region is wherever the DNA is floating within the cytoplasm. The ribosomes are in the cytoplasm, but they are too large to be dissolved in the cytoplasm. Inclusions are substances that the cell is accumulating, and once the substance gets to be so high in concentration that the substance can no longer be dissolved in the cytoplasm, it will precipitate out of the cytoplasm, and then we can see the inclusion body in the cell. When it is dissolved in the cytoplasm, you wouldn't see it. It'd just be part of the cytoplasm. The nucleoid region, or the nuclear area, is wherever the single chromosome DNA is located. There is no nuclear membrane to separate the cytoplasm from the nuclear area. It's wherever the DNA happens to float in the cytoplasm. The ribosomes are made up of protein and RNA, and they are so large that they do not go into solution of the cytoplasm, and you can actually see them in the cell with an electromagnetic microscope. There are these little dots there, or the dots here. Ribosomes are important for the cell because they are the site of protein synthesis. They are made up of both protein, we can call it ribosomal protein, and RNA, we call it rRNA or ribosomal protein. The prokaryote ribosomes are a little different than the eukaryote in that they're smaller. They're 70s in size. I may talk about the eukaryotes, or maybe I won't, I don't remember. But the eukaryote ribosomes are 80s in size. You don't need to know about the S, but that's a unit that microbiologists and biochemists have for discussing ribosomes. The 70s is where the ribosome moves to in an ultracentrifuge gradient. And the 80s ribosomes are a little bigger, so they're a little bit lower in the ultracentrifuge gradient. You don't need to know that there are subunits that make up the ribosome. Inclusions are reserved deposits that the cell is accumulating within the cytoplasm, and it accumulates to such levels that the deposit precipitates out of solution. There are different examples of inclusions. You don't need to know the different names. You just need to know that there are different examples of inclusions. That's all you need to know. Here we're looking at some inclusions, magnetosomes, sulfur granules. Let me shrink that just a little. Where the cell is accumulating sulfur at such high levels that the sulfur starts precipitating and forming sulfur inclusions called sulfur granules. This cell is obviously going to metabolize sulfur. And then metachromatic granules. There's 3 of them in the cell. Let me blow that up a little. You can see the cell outlined in yellow there. Any question about inclusions? An endospore is a specialized resting cell produced by certain species, mainly the members of Bacillus and Clostridium family. Endospores are highly resistant to desiccation, heat, chemicals, radiation. They can survive in conditions where the vegetative cell cannot. The vegetative cell is the parent cell, which makes the endospore. So when the environment starts deteriorating, the parent cell will start making an endospore. An endospore is a survival structure. It is not a reproductive structure. One cell will make one endospore, and as the environment deteriorates further, the parent cell will die. One cell making one cell is not reproduction. Once the endospore begins forming, that's called sporulation or sporogenesis, where the endospore begins forming. Once the environment becomes more favorable to the cells to grow and whatnot, go through their normal metabolism, the cell will go through germination. The endospore will germinate, returning to a vegetative cell, a cell without the endospore, and the vegetative cell can grow and reproduce and go through normal metabolism. The endospore is a resting cell. It does have a little bit of metabolism, but very, very little. Its purpose is just to survive the bad environmental condition. Here we're seeing an endospore being made inside this cell. And right there you can see all of these cells, the white, let me blow that up a little more. The white thing there is an endospore being made within that cell. And there's a lot of them being made here. Oops, didn't mean to do that. All right, endospore formation happens when the cell goes through division. It will divide into two DNA molecules, which will move to the opposite ends of the cell. Unlike normal cell division, to make an endospore, the cell does not divide in two evenly. It divides into a smaller compartment and a larger compartment. The plasma membrane starts to surround the DNA, and then the plasma membrane actually becomes two membranes around the endospore. The plasma membrane or the peptidoglycan layer forms between the two membranes. And then the spore coat forms on the outside of the cell, and outside of the endospore, I should say. And then the environment deteriorates further, and then the parent cell, which was this portion of the cell here, then dies, and that will release the endospore. Any question on endospore formation? We have a little video, but it's not very good. There it goes. Just took a while.
Showtime.
So what happened to your face? You look terrible.
Cell forming an endospore. All right, any questions about any of that? All right, this slide is showing you the cell structure seen in an animal cell and a plant cell. So for eukaryote cells, you can note that there are some different structures between plant cells and animal cells. For one, the chloroplast is found in photosynthetic plant cells, but not in animal cells. The chloroplast is not found in all plant cells. Like in the root of a plant, you won't find any chloroplasts, only the photosynthetic cells. You can find a central vacuole in a plant cell. You do not find a central vacuole in an animal cell. The central vacuole is the largest organelle in a plant cell. The largest organelle in an animal cell is the nucleus. You can find vacuoles in an animal cell, and you can find vacuoles in a plant cell. So many of them, like the perioxysomes, the mitochondrion, the Golgi complex, you can find these in both a plant cell and an animal cell, but not all of them. There is one that is found in an animal cell and not found in a plant cell. Let me see if I can find it here. There it is. The centrosome, containing the centriole and the pericentral material, shown right there. It's involved in helping the cell form the spindle in mitosis and meiosis. Animal cells have them, the centrosome, but plant cells do not. Plant cells do form a spindle in mitosis and meiosis, but they don't have a centrosome, which helps make the spindle. Any questions about any of that? Many of the organelles are the same, like the mitochondria, the Golgi apparatus, both plants and animal cells, the nucleus in both plant and animal cells. Here are the prokaryotes and the eukaryotes. Let me see if I can blow this slide up. So prokaryotes are smaller than eukaryotes typically, about 1/10 the size. When you're talking about the average cell, that's pretty good for an average cell, because the range it does get larger than 2 micrometers. Although 0.02 is really, really small. As far as I know, that's a non-viable prokaryote cell. And then it depends on obviously what cell we're talking about, but the eukaryotes are generally about 10 times larger than the prokaryotes. The nucleus is absent in the prokaryote, present in the eukaryote. The membrane-bound organelles are absent in the prokaryotes. They are present in the eukaryotes. Flagella are found in both, although they differ. Glycocalyx is found in both, but not all cells have a glycocalyx. The cell wall is usually found on a prokaryote, but not all of them. And a cell wall is found on some eukaryotes, like plants and fungi and many protists, but is absent on an animal cell. Very differ in the cell wall. The prokaryote has a chemically complex cell wall. Bacteria have peptidoglycan. Eukaryotes have a very simple cell wall, and they never have peptidoglycan in their cell wall. The plasma membrane differs. Usually there's no carbohydrates and lacks steroids, although there is a family that does have sterols in it. I'm talking about bacteria. And then there's a cholesterol, a sterol in eukaryote cell walls. Not cell walls, the cell membrane. I might have said that wrong for the prokaryotes. We're talking about the cell membrane there. Cytoplasm, this is a little old. There is a cytoskeleton in the prokaryotes, but it's simple compared to the cytoskeleton in eukaryotes. Very complex cytoskeleton in eukaryote cells. There's also cytoplasmic streaming, where the liquid portion, the cytoplasm, moves around in the eukaryote cell. There is no cytoplasmic streaming in the prokaryotes. The ribosomes are smaller in size in the prokaryotes, 70S. They're larger in size, 80S, in the eukaryote. We'll talk about the organelles of eukaryotes later. DNA differs too. In a prokaryote, the DNA is usually in one chromosome, which is circular. So where the chromosome begins, right here, is the same place where it ends. It's a circle. Whereas the chromosomes in eukaryotes are generally many, and then they're linear. So where the chromosome begins, right here, will differ where the chromosome ends, right here. In cell division differs too. Most prokaryote cells divide by binary fission. Eukaryote cells divide by mitosis. Reproduction also differs. There is no sexual recombination in prokaryotes, although they can transfer plasmids, which would be similar. In the cell divides by binary fission, usually by parthenogenesis. And in most eukaryote cells, they reproduce by meiosis, sexual reproduction and meiosis. Any question about any of that? Let me state that if the eukaryote cell moves, it can move by one of three structures: cilia, flagella, and then pseudopods. If you remember, prokaryotes only move by one structure, and that would be a flagella. Any question about any of that? All right, let's move on to the endosymbiotic theory. All the other material in this chapter should be reviewed for you, and I'm starting to run out of time, so I'm not going to cover that material. I usually don't get too much of it. It should be review for material for you, so you should be able to go through the slides and understand it on your own. But I need to talk about the endosymbiotic theory. This is a theory to explain the evolution of mitochondria in chloroplasts in eukaryotes. How did these organelles originate? Well, the chloroplasts and the mitochondria differ from all other organelles in that, first of all, they have two membranes, an inner and outer membrane of that organelle, and they also have DNA molecules within them and ribosomes within them. No other organelle has these. How did these organelles come to be? Well, it's thought, let me make up paint here. It is thought that in the evolution of eukaryote cells, a long time ago, before we had true eukaryotes, we had a proto-eukaryote, which was a very large cell. I'm just going to go PE to mean proto-eukaryote. Oh, I guess I can spell it. This is a cell before becoming a eukaryote. It had no mitochondria or chloroplasts. It may have had other membranes, but it was a large cell. And this large cell engulfed a small cell that was a bacteria. Yeah, that's a little thin there. how it engulfed it was by pseudopods, and let me see if I can erase this. Oh, there it is. There's the brush. Bring that closer to the cell. Doesn't matter what this shape is. Pseudopods grew out of the proto-eukaryote. Further. And they grew further until the pseudopods touched each other. And what happens when a membrane touches another membrane is sometimes the membrane can fuse. Now we have the bacteria inside the cell. Any question about that? Well, this bacteria could engage in aerobic respiration. Meaning it can make from one glucose, it could make 36 ATP. Oops, I don't have that right. Actually, it should be 38. The proto-eukaryote could not engage in aerobic respiration. It could engage in fermentation. I guess it's this one right here. Making 2 ATP. Oop. Per molecule of glucose. Now, for whatever reason, the proto-eukaryote did not eat the bacteria that engulfed right away. And some of the glucose in the cytoplasm, let me make that here, moved by diffusion into the vacuole containing the bacteria and then moved into the bacteria. And obviously when it's moving across the membrane, it's not going to move by diffusion. That will be by probably active transport. The point is that some of that glucose moved from the cytoplasm of the proto-eukaryote into the cytoplasm of the bacteria. The bacteria could make 38 ATP per molecule of glucose. And for whatever reason, maybe because life was early back then, the bacteria gave some of its ATP to the proto-eukaryote. It probably was not 38 ATP, but was much more than 2 ATP. We don't know the exact number, But the point is that the glucose moved from the proto-eukaryote to the bacteria, which made made ATP out of it, and then shared some of the ATP with the proto-eukaryote. The proto-eukaryote was very confused because it only lost one glucose molecule, and now it gained a whole bunch of ATP. The proto-eukaryote figured out, hey, this is pretty good. I feed glucose to the bacteria. I make a lot more ATP from that glucose than I can make. And so the two started living together in symbiosis. The proto-eukaryote with the bacteria inside it. The proto-eukaryote fed the bacteria glucose, and then the bacteria made lots of ATP for the proto-eukaryote, much more ATP than the proto-eukaryote could make from a molecule of glucose. The two have lived together for so long that now we no longer find the bacteria living on its own. We only find it living within the proto-eukaryote. And the bacteria became the mitochondria of the proto-eukaryote, which now we're going to call the eukaryote cell. Any question about that? So the two now live together. How do I word that? That's the theory of how mitochondria evolved. It's also how chloroplasts evolved. We'll talk about chloroplasts in just a few minutes. Well, what's the proof that this theory may have occurred? Well, first, if a bacteria was engulfed by a proto-eukaryote, there should be two membranes around the bacteria, the cell membrane and the vesicle membrane around the bacteria when it was engulfed by the cell. And we do find two membranes around the mitochondria, the inner mitochondrial membrane and the outer mitochondrial membrane. Here. We also have some other evidence for the theory occurring, and that is we have some protists which can take on other cells like this. It looks like a paramecium has taken up a bacterium, and then the paramecium feeds the bacteria, gives the bacteria a home, and then the bacteria engages in photosynthesis, why it's kind of greenish here, and then shares the sugar that the bacteria makes with the paramecium. So the paramecium can live together with another cell that it engulfs, and then they live in symbiosis. So we do see that happening. Also, there's a family of amoeba which lack mitochondria. And these amoeba go out once they are newly formed by probably by mitosis, or maybe meiosis, I don't know. The amoeba go out and then they swallow a bacteria, engulf a bacteria, bring it inside the cell, and then feed the bacteria glucose. And then the bacteria make ATP and then share out of the glucose and then share it with the amoeba. So we actually find some amoeba that lack a mitochondria. And what they do is they engulf a bacteria and then have that bacteria inside them serve as a mitochondria, making the amoeba ATP. And the reason why the amoeba does it is because the bacteria can make much more ATP from aerobic respiration, and the amoeba can only get 2 ATP from each molecule of glucose by fermentation. There's some other evidence. When we look at the DNA of a prokaryote cell, generally there's one molecule of DNA, it's circular. A eukaryote has different DNA. Usually there's more than one chromosome. The DNA are linear. When we look at the DNA of the mitochondria and the chloroplast, they are circular molecules of DNA, and there's only one molecule of DNA as well. When we look at the presence of histones, histones are a major protein that the DNA wraps around in a eukaryote cell. There are no histones in a bacteria. There are no histones in the mitochondrial DNA or the chloroplast DNA. When we look at the first amino acid made in protein synthesis, in a eukaryote, it's always methionine. In a prokaryote cell, it's a related amino acid, but it's a different amino acid. It's formylmethionine. When we look at the first amino acid made in protein synthesis in the mitochondria of the chloroplast, it's always formylmethionine. When we look at the ribosomes of a prokaryote, they're 70s in size. When we look at the ribosomes of a eukaryote, they're 80s in size. When we look at the ribosomes of the mitochondria and the chloroplast, they're 70s in size. The prokaryote cell splits mostly by binary fission. The mitochondria and the chloroplast split by binary fission. Not mostly, but by binary fission. And then the eukaryote cell, almost all eukaryotes split by mitosis. So their cellular growth and reproduction differ too. And then the strongest evidence that the mitochondria and the chloroplast are not eukaryote membrane-bound organelles, that they are a prokaryote cell that became the mitochondria and the chloroplast living inside the eukaryote cell. When we look at the DNA synthesis DNA sequence of the mitochondria and the chloroplast, we find that they fall within the domain bacteria, not within the sequence of domain eukaryote. Any question about any of that? All right. So that's the theory of endosymbiosis, where in an animal cell, a long time ago, we had a proto-eukaryote cell, or ancestor eukaryote cell, that engulfed a bacteria. And then the two started living together. The proto-eukaryote feeding the mitochondria, giving it a home, and then the bacteria making ATP for the eukaryote cell. And that bacteria later became the mitochondria in the eukaryote cell. In plants, this happened, same thing. However, another cell got engulfed by the plant cell. So the plant cells engulfed both the bacteria and another bacteria. One bacteria became the plant cell's mitochondrion, and the other bacteria became the plant cell's chloroplast. Any question about any of that? All right, that's the theory of endosymbiosis. Let me go ahead and back up and talk about the endomembrane system. This is a system of membrane-bound organelles that we see in a eukaryote cell. For both proteins and lipids, they generally begin life in the endoplasmic reticulum. It is true that proteins can be made in the cytoplasm as well as in the nucleus, but many proteins are made in the rough ER, and it's called rough ER because the endoplasmic reticulum looks rough because there are ribosomes attached to the rough ER. Any question about any of that? Making the ER look rough from the ribosomes. There's also smooth ER, which is endoplasmic reticulum lacking ribosomes. The smooth ER do not make proteins, they make lipids. The proteins that are made by the rough ER get packaged up in the ER, made into a vesicle which breaks off of the rough ER, and then that vesicle is transported by a transport vesicle to the Golgi apparatus. The vesicle actually binds to the Golgi apparatus, and then the two membranes fuse so that the transport vesicle, becomes a portion of the Golgi apparatus. The proteins are put in this side of the Golgi apparatus, where they then become modified, like chemically modified. You can change the DNA, not the DNA, you can change the protein. Like you can remove certain amino acids, you can put methane groups on the amino acid. You can do other alterations of the protein in the Golgi. And then the protein moves from this side of the Golgi apparatus to this side of the Golgi apparatus. And then the proteins are packaged up. And then a vesicle buds off of the Golgi apparatus from this side of the Golgi apparatus. And then the vesicle transports what's inside it, meaning the proteins that have been modified, to wherever the proteins need to go in the cell. And that could be, the proteins could be brought to the cell membrane, and then the proteins can be dumped outside of the cell membrane, such as in teenagers when they're making hormones. Their cells dump them out by using the endomembrane system. But the protein will be transported by a transport vesicle to wherever the protein needs to be. It could put it in, if you make lysozyme and other components of the lysosome, the vesicle can fuse to the lysosome or become a lysosome, and then the lysosome can fuse to a phagosome and then dump the contents of the lysosome inside the phagosome to digest the products of the phagosome. If the transport vesicle just moves to a location and then stops moving and then just stores the contents of the vesicle, we no longer call it a transport vesicle because it's not transporting. We call it a vacuole because it's not moving and it's just storing contents. So a vacuole is the same as a transport vesicle, except the vacuole is storing things and it's not moving. The transport vesicle is actually moving things around in the cell, transporting it to where they should go. And that's the endomembrane system involving the ER, which happens for the proteins I talked about, but it also happens for the lipids. And then the contents from the ER are brought to the Golgi apparatus for modification, sorting, and then they come out of the Golgi apparatus as a vesicle once again, transport vesicle, which moves the modified protein or modified lipid. We haven't talked about lipids, but just say that the same thing happens with lipids to where they should be in the cell. Any questions about the endomembrane system? All right. If there's no questions, I'm going to end this lesson a little bit early, and I will see you at 6:30 for the lab. Bye.